Hello and welcome everyone. Myself Mijanur Rahaman, and today my topic of discussion is regulation of respiration. So today we'll start, and today we'll try to cover all the parts, but as much as possible we can do that. So first our topic will be introduction part. Then we will try to cover the nervous mechanism, and if possible we will try to cover the chemical mechanism. Okay. So let us start. First of all, introduction. So respiration is a reflex process. So respiration is what? It is a reflex process, but it can be controlled voluntarily for a short period of about 40 seconds. However, by practice, breathing can be withheld for a longer period. It is possible. At the end of that period, the person is forced to breathe. Now, the respiration is subjected to variation even under normal physiological condition. For example, emotion and exercise increase the rate. What increases? The emotion and exercise increases the rate and force of respiration, but altered pattern of respiration is brought back to normal within a short time by some regulatory mechanism of the body. Normally, quiet breathing occurs because of two regulatory mechanisms. One is nervous or neural mechanism, and one is chemical mechanism. So first our discussion will be on nervous mechanism. Now the nervous mechanism that regulates the respiration includes respiratory centers, afferent nerves and efferent nerves. Okay. Now first of all the respiratory centers. Respiratory centers are the group of neurons which controls the rate, rhythm and force of respiration. Now what is respiratory centers? Respiratory centers are group of neurons which control the rate, rhythm and force of respiration, and these centers are bilaterally situated in reticular formation of the brainstem, as you can see in the diagram. So what is respiratory centers? Respiratory centers are the group of neurons which control the rate and rhythm and force of respiration. Now respiratory centers are the group of neurons which control the rate, rhythm and force of respiration, and these centers are bilaterally situated in the reticular formation of the brainstem, which is described in this figure. Now depending upon the situation of the brainstem, the respiratory centers are classified into two groups. One is medullary centers consisting of dorsal respiratory group of neurons and ventral respiratory group of neurons. So now there are two centers in case of respiratory center. Respiratory center have the medullary center and pontine center. The medullary center is consist of the dorsal respiratory group of neuron and ventral respiratory group of neuron. But the pontine center are consist of the apneustic center and pneumotaxic center. So first start with the medullary center. Now the medullary center have the dorsal respiratory group of neurons, and dorsal respiratory group of neuron are diffusely situated in the nucleus of tractus solitarius, which is present in the upper part of the medulla oblongata. So dorsal respiratory group of neurons are diffusely situated in the nucleus of tractus solitarius, which is present in the upper part of the medulla oblongata, and these neurons are collectively called inspiratory center. So what is dorsal respiratory center? Dorsal respiratory group of neurons are diffusely situated in the nucleus of tractus solitarius, which is present in the upper part of the medulla oblongata, and usually the neurons are collectively called the inspiratory centers. You can see there the vagus nerve, the chemoreceptors, the ventral group neurons, the dorsal group neurons, the apneustic center, pneumotaxic center. So we have discussed first the dorsal respiratory center or inspiratory center. Here it is in the pink color. You can see that. Now Here the dotted line and the solid line are differentiating one another. Dotted line are inhibition mechanism and the solid green line are the stimulatory mechanism. We will discuss in the later part of the video. Now all the neurons of dorsal respiratory group are inspiratory neurons and generate inspiratory ramp by their virtue of their autorhythmic property. Now all the neurons of the dorsal respiratory group are inspiratory neurons and generate inspiratory ramp by the virtue of their autorhythmic property. Okay, now the function. Dorsal group of neurons are responsible for the basic rhythm of respiration. Now the experimental evidences. Now what are the autorhythmic property? We will discuss in the table later on. Now function is the dorsal group of neurons are responsible for the basic rhythm of respiration. Now experimental evidences are electrical stimulation of these neurons in animals by using needle electrode causes the contraction of inspiratory muscles and prolonged inspiration. Now we will discuss about the table or chart. Now there are dorsal group and ventral group. Medullary centers. Situation. Dorsal group is situated diffusely situated in the nucleus of tractus solitarius, but the ventral group is present in the nucleus of ambiguous and nucleus retroambiguous. Now type of neuron. The dorsal is inspiratory and the ventral is inspiratory and expiratory both. Function. The dorsal is always active and it generates inspiratory ramp and it has autorhythmic property. And in case of ventral, inactive during quiet breathing and active during forced breathing. Now start with the ventral group of neurons. Ventral group of neurons is situated ventral group of neurons are present in the nucleus ambiguous and nucleus retroambiguous, and these two nuclei are situated in the medulla oblongata anterior and lateral to the nucleus of tractus solitarius, and earlier the ventral group of neurons are collectively called expiratory centers. So in the pink color you can see the dorsal group of neurons, in the green color you can see the ventral group of neurons. And the ventral respiratory group has both inspiratory and expiratory neurons, and the inspiratory neurons are found in the central area of this group. In the dotted line and the green line you have seen that the solid green line is stimulation, dotted red line are inhibition, and in case of the ventral group it is both inspiratory and both expiratory, but in case of dorsal it is only and only inspiratory. Now inspiratory neurons are found in the central area of the group. You can keep in mind this point, central area. But expiratory neurons are in the caudal and rostral areas of this group. So this is an important question. The inspiratory group of neurons are found in the central area of this group, but the expiratory neurons are present in the caudal and rostral areas of this group. Now what are the functions? Function are normally the ventral group neurons are inactive during the quiet breathing and become active during the forced breathing. But during the forced breathing, these neurons stimulate both inspiratory muscle and expiratory muscle, and some experimental evidences in case of this. The electrical stimulation of the inspiratory neurons in the ventral group causes the contraction of inspiratory muscles and prolonged inspiration. Now, stimulation of expiratory neurons causes the contraction of expiratory muscles and prolonged expiration. Now, we will discuss about the pontine center. Pontine center, it has one is apneustic center. The apneustic center is situated in the reticular formation of the lower pons. You can see in the blue color. The apneustic center is situated in the reticular formation of the lower pons, and its function is to increase the depth of the inspiration by acting directly on the dorsal group of neurons. Now some experimental evidences. The stimulation of apneustic centers causes apneusis, and apneusis is an abnormal pattern of respiration characterized by the prolonged inspiration followed by short and inefficient expiration. Now the pneumotaxic center. Pneumotaxic center, it is present in the or it is situated in the dorsolateral part of the reticular formation in the upper pons, and it is formed by the neurons of medial, parabrachial and subparabrachial nuclei. So pneumotaxic center is situated in the dorsolateral part of the reticular formation in the upper pons, and it is formed by the neurons of medial parabrachial and subparabrachial nuclei, and the subparabrachial nucleus is also called the ventral parabrachial or collicular fuse nucleus. Now what is the function of pneumotaxic center? The primary function of pneumotaxic center is to control the medullary respiratory centers. What is the primary function of the pneumotaxic center? Is to control the medullary respiratory centers, particularly the dorsal group of neurons, and it acts through the apneustic center, and pneumotaxic center inhibits the apneustic center so that the dorsal group of neurons are inhibited. Because of this, the inspiration stops and expiration starts. Thus, pneumotaxic center influences the switching between the inspiration and expiration. So, pneumotaxic helps to switch in between the inspiration and expiration. And pneumotaxic center increases the respiratory rate by reducing the duration of inspiration. Now, some experimental evidences. Stimulation of pneumotaxic centers does not produce any typical effect except slight prolongation of the expiration by inhibiting the dorsal respiratory group of neurons through apneustic center. Destruction of or inactivation of pneumotaxic center results in apneusis. Now, if you repeat this part again, today what we have discussed. First of all, we have introduced what is respiration. Respiration is nothing but a reflex process. It can be controlled voluntarily for a short period of about 40 seconds, and however by practice a breathing can be withheld for a long and longer period, and at the end of the period the person must force to breathe. The respiration is subjected to variation even under normal physiological condition. For example, the emotion and exercise increases the rate of force of respiration, but the altered pattern of respiration is brought back to normal within a short time by some regulatory mechanism in the body. Now, normally the quite regular breathing occurs because of two regulatory mechanisms. One is nervous or neural mechanism, one is chemical mechanism. In case of nervous mechanism, the nervous mechanism that regulates the respiration includes respiratory centers, afferent nerve, efferent nerve. And the respiratory centers are what is now respiratory centers? Respiratory centers are nothing but a group of neurons which control the rate, rhythm and force of respiration. So if someone asks you to what is respiratory center, then you can give the answer like that. The respiratory centers are nothing but a group of neurons which can control the rate, rhythm and force of respiration. And these centers are bilaterally situated in the reticular formation of the brainstem, and depending upon the situation of the brainstem, the respiratory centers are classified into two groups. One is medullary center, one is pontine center. And in case of medullary center, you can find there are two parts. One is dorsal respiratory group of neurons, which is only found in case of inspiration, and the ventral respiratory group of neurons, which are acting both time inspiration and expiration. Now the next pontine center, there are apneustic center and pneumotaxic center. Now first the medullary center. In case of medullary center, first of all the dorsal respiratory group of neurons. It is situated dorsal respiratory group of neurons are situated diffusely in the nucleus of tractus solitarius, which is present in the upper part of the medulla oblongata. So where it is present? It is present in the upper part of the medulla oblongata in the nucleus of tractus solitarius. You can see in the diagram also. Now, usually these neurons are collectively called the inspiratory centers, and all the neurons of the dorsal respiratory group are inspiratory neurons and generate the inspiratory ramp by the virtue of their autorhythmic properties. So, these type of groups, the dorsal group of neurons, have an important property of autorhythmicity. Now, what are the functions? Dorsal group of neurons are responsible for the basic rhythm of respiration. Now some experimental evidences. Electrical stimulation of these neurons in animals by using needle electrode causes contraction of inspiratory muscle and prolonged inspiration. Now, if we start the ventral respiratory group of neurons, it is present or it is situated in the nucleus ambiguous and nucleus retroambiguous, and these two nuclei, the nucleus ambiguous and the nucleus retroambiguous, are situated in the medulla oblongata. So the dorsal group of neurons are present in the tractus solitarius in the upper part of the medulla oblongata, and the ventral respiratory group of neurons, these are also present in the medulla oblongata, and these are present in the two nuclei. One is nucleus ambiguous and one is nucleus retroambiguous. And anterior and lateral to this nucleus are tractus solitarius. Earlier, the ventral group of neurons are collectively called expiratory centers, but now we found that in the past we only know that the ventral group of neurons are known for the expiratory group of muscle, but in the modern era, we come to know that the ventral respiratory group has both inspiratory and expiratory neurons. In the past, we know about the only expiratory neurons. In the present, we know about the inspiratory and the expiratory neurons both. Now, inspiratory neurons are found in the central area of this group, but the expiratory neurons are found in the caudal and rostral areas of this group. So, if someone asks you where the inspiratory neurons of ventral respiratory group of neurons are present, you can say that it is present in the central area of this group. But in the expiratory neurons, it is present in the caudal and the rostral areas of this group. Now, what are the function? Function is normally ventral group neurons are inactive during the quiet breathing and become active during forced breathing. And during forced breathing, these neurons stimulate both inspiratory muscle and expiratory muscle. Some experimental evidences. Electrical stimulation of the inspiratory neurons in the ventral group causes contraction of inspiratory muscle and prolonged inspiration, and stimulation of expiratory neurons causes the contraction of expiratory muscle and prolonged inspiration. Now, if we see the difference between two medullary centers, that the first feature, in case of dorsal group, it is diffusely situated in the nucleus of tractus solitarius, and by the ventral group, it is present in the nucleus of ambiguous and nucleus retroambiguous. Type of neuron: dorsal is inspiratory, ventral is previously known as expiratory neurons, but now it is known as inspiratory and expiratory neurons. Now, function: dorsal is always active and it is generate the inspiratory ramp and has autorhythmic property. But the ventral group, it is inactive during the quiet breathing and active during the forced breathing. Now, the pontine centers. In case of pontine centers, the first one is the apneistic center, which is situated in the reticular formation of the lower pons, and its function is to increase the depth of the inspiration by acting directly on the dorsal group of neurons. Some experimental evidences. The stimulation of apneistic center causes apnesis. And apnesis is an abnormal pattern of respiration characterized by the prolonged inspiration followed by short, insufficient expiration. Now, the pneumotaxic center, which is situated in the dorsolateral part of the reticular formation in upper pons, and it is found by the neurons of medial, parabrachial, and subparabrachial nuclei. And the subparabrachial nucleus is also called the ventral parabrachial or follicular fuse nucleus. The function of the pneumotaxic center is to control the medullary respiratory centers, particularly the dorsal group of neurons. It acts through the apneistic center, and pneumotaxic center inhibits the apneistic center so that the dorsal group of neurons are inhibited. Because of this, inspiration stops and expiration starts. Thus, the pneumotaxic center influences the switching between the inspiration and expiration. It switches off the apneistic center, which further switches off the dorsal group of neurons, which further stop the inspiration and start the expiration. Thus, the inspiration and expiration switches between them. Now, pneumotaxic center increases the respiratory rate by reducing the duration of inspiration. Now, experimental evidences: the stimulation of pneumotaxic center does not produce any typical effect except slight prolongation of the expiration by inhibiting the dorsal respiratory group of neurons through apneistic center, and the destruction or inactivation of pneumotaxic center results in apnesis. Now you see again the diagram. Here is the lungs, the vagus nerve, which is going the inhibitory mechanism, the ventral group of neurons, the dorsal group of neurons, the apnestic center, the pneumatic center, the now the dorsal group of neurons are for the inspiratory mechanism, the ventral group of neurons for the inspiration and expiration both. First, the one person is taking inspiration or taking the breath with the help of the dorsal group of neurons. Then, when the inspiration is complete, the pneumatic center signals the apnestic center negative signal that you must stop so that the dorsal group also stop and the inspiration must stop and expiration starts. Now by this mechanism the inspiration and expiration switches in between them. So this is our today's short topic. Today I have thought that we will complete more than this, but we thought that it is not good for all of us. It is much better to complete lower portion but complete it as it is possible with good quality. So thank you so much for watching the entire video. If you have any query or any doubt, you can comment down in the comment section, and thank you so much for watching the entire video.